So can we start?
Yeah. Can we be live, Ananya?
Going live, ma'am.
Okay. A very good afternoon, everyone. Speaker of the session, Dr. Seema Sangwan, scientist, Indian Institute of Agriculture Research, Dr. Veda Krishnan, Secretary INYAS, Dr. Aarti Kumari, State Coordinator, Microbiological Society of India, Professor Sunita Agarwal, State President, Microbiological Society of India, and Dr. Subhash Yadav and other members and all dear participants. I, Dr. Kalpana Nagpal, Coordinator, Delhi NCR Chapter, welcome you all on this Sustainability Lecture Series, which is jointly organized by INYAS NCR Chapter, Institute of Home Economics, Department of Microbiology, Delhi University, and Microbiologist Society, India. With a vision to make this planet sustainable, UN has put forward its Sustainable Development Goals, and India is a key partner playing a major role in this global agenda. So as per our Honorable Prime Minister, Shri Narendra Modi ji, achieving sustainability will open to a world fewer challenges and, of course, greater hope. Hence, science, technology, and innovation have a great role to play in it. INYAS, in collaboration with University of Delhi and Microbiological Society of India, here are organizing this lecture series to have a dialogue on different facets of sustainability with the major theme, role of science in sustainable development and Indian perspective. So this is the second lecture in the series. Today, Dr. Seema Sangwan will talk about the role of microbes as fertilizers, their types, their importance and benefits, and of course, how they are leading partners towards sustainability. Now, may I request Professor Sunita Agarwal, State President, Microbiological Society of India, to brief all the audiences about the today's speaker of the session. Professor Sunita, please.
Thank you, Dr. Kalpana. Good afternoon, everyone, and welcome to the second talk of the Sustainability Lecture Series on a very relevant topic of today, biofertilizers, types, importance, and their benefits by Dr. Seema Sangwan. Dr. Seema is presently working as a scientist in Division of Microbiology, Indian Agriculture Research Institute. She did her PG in microbiology from Kurukshetra University and PhD in dairy microbiology from National Dairy Research Institute, Karnal. Dr. Seema got a Young Scientist Award at National Seminar on Recent Advances in Development of Fermented Food at Banaras Hindu University. She has published around 18 research papers in reputed national and international journals. We welcome you once again, Dr. Seema, to today's webinar and extremely thankful for accepting our invitation and taking out time from your busy schedule. Now, Dr. Seema, please, dice is yours. Dr. Seema, please.
Thank you, Dr. Sunita ma'am, and thank you, Kalpana ma'am. I'm very glad that I got this opportunity to share our work, what we are doing, and what is really important for this earth now, as our Prime Minister and everybody globally, they are saying that what we should do in towards, especially in the area of agriculture. So I would like to start my talk now. Please allow me to share my PPT. Yes, give me 1 minute. So is it visible, ma'am?
No, not yet.
Not yet. Okay.
You can unshare and reshare the screen.
Yeah, I'll try again. I'm sorry, there is some issue.
No problem. Take your time.
Uh,
Just a submission to all the students. If you find any difficulty or doubt, please note down your queries or you can write to us also. So at the end, we shall be taking the questions from your end. Thank you.
Dr. Kalpana, our director ma'am is here, Dr. Renu Arora, Professor Renu Arora.
Yes,
Uh,
ma'am.
Hello, hi everybody. Sunita, hello, how are you?
I'm
Welcome,
fine.
ma'am.
Thank you.
Good afternoon, ma'am.
Good afternoon, everybody.
Submission to all the
Sunita,
students.
it will not be possible for me to attend the entire program, but
No, no problem. No problem. Yeah, I know.
yeah, so very delighted that today this lecture 2 series of sustainability is initiated by INYAS and MSDI and Department of Microbiology IHE. And Dr. Seema, I believe, is this resource person.
Yeah, Dr. Seema is
Yeah.
there.
Yes, ma'am.
Dr. Seema, thank you so much from IARI. And
Thank
yeah,
To
are
you
going to
too,
talk.
ma'am, to give this
Hello.
opportunity.
Yeah, and thank you for taking out the time, especially on Saturday. And a very important area is being going to be covered, that is microbes and its benefits. Am I right?
Yes, ma'am.
Yeah. So wishing all of you the best of luck, and I'm sure everybody would benefit. And we have done a lot of work with IRI, senior scientist Dr. JP Sharma. You must be knowing him.
Yes, ma'am. Yes, ma'am.
या हमने बहुत काम किया कि अपने टाइम पे 15-20 साल पहले lot of extension activities and training program and entrepreneurship. It really used to be a fun having these programs over there. So conveying my best wishes to the students as well as to the staff. And जितना मैं हो सकेगा सुनीता बीच-बीच में join करूंगी है.
हाँ जी. Thank you.
Thank you so much. Okay,
Ma'am,
best of
just
luck.
can you give me a minute? I will try from the other computer.
Yeah.
There is some issue I'm getting. Just give me a minute, please.
So, Dr. Kalpana, we have a old association with IARI.
Okay.
Even from my student days, I used to go there for the experiments and all.
Dr. Seema, if you are finding any difficulty, you can share the presentation, and Ananya, if you can share on her behalf, if possible.
Sure, ma'am, I may.
Yes,
Dr.
ma'am.
Seema, can you email your presentation in that case?
Yes, ma'am. Please
Yeah.
give me the mail ID to my.
Okay. Sure. I just emailed you yesterday, so you can revert there also.
Okay. I'm sending the presentation, ma'am, to that email only through which we were communicating. So within a 1 or 2 second, it will be.
Yeah, let me refresh.
How many students have joined, Sunita?
Total 38 participants are here. Some may have joined through the YouTube also.
YouTube also, yeah. So they are from all the years, second year, third year, first year?
First year, so they have the exams
The
going
exams
on.
are going on. Okay, exams are going on. Yeah.
So only second year and third year here.
Okay, so they can see the recording later also, no problem.
I have sent it, ma'am. Please refresh.
Yeah, I just refreshed but could not see. Give me another second. Is it showing sent from your end?
Yes, yes. Yes, ma'am.
Okay.
Technology is good, but sometimes it creates a huge issue.
It's okay. Once it will be on pace, I think.
Dr. Seema, since how long have you been associated with IRI?
Ma'am, I joined, my first posting is in IRI and I joined in 2016.
It's 2016.
Yes,
Acha.
yes,
Where were
yes.
you before that?
No, this was my first posting only.
Acha, this is the
Dr.
first
Seema,
posting
sorry
only.
to
Oh,
interrupt. Did you get my email as a test email? Because I just reverted you.
I got it, her PPT.
Presentation I did not get. Just to confirm whether you use the same email address.
I got it, I think. Let me see. It's, yeah, I got the mail from Seema and got the PPT also.
Okay.
Shall
Can
I,
you share?
shall I share?
Yeah. Yes, please.
are you
Thank
sure, Seema?
Please, no problem, I think. I think you have to give the access to me because I'm not able to share.
Ananya, can you please make a
Uh,
post?
The access is updated, ma'am. Anyone can share.
Okay. Ma'am, are you able to do it now?
No, I'm not able to do it. There's some problem, I think. Shall I send the PPT to you again?
Here on my system also, I'm getting some issue.
So in the meantime, just give me a moment to brief about INYAS. INYAS is Indian National Young Academy of Sciences, and it was founded by INSA in December 2014. Its vision is to promote science education and networking among the young scientists. And this journey of so many years has been incredibly so far with full of new learnings and many achievements to its credit. So at first, it started with 20 young scientists as a founding batch member. And with respect to time, we are continuously growing and keeping ourselves dynamic. The committed members, which are contributing towards the various objectives of INYAS, they are from various disciplines like physics, chemistry, biology, mathematics, engineering, even earth and atmospheric sciences, and medical sciences. So, with the same theme and objective to promote science, we have a membership of 5 years. That's how we always keep ourselves young. So, those who complete their tenure of 5 years become the alumnus of INYAS. So I think, can we start the presentation now if it is?
No, I'm not able to share. I have mailed you the same PPT. You just check, Kalpana.
Okay, let me check. Yeah.
Have you got it?
Yes. Ananya, can you please share?
Yes, ma'am. If you can please
Yeah.
forward it to me.
Yeah, done. Please check.
Email or WhatsApp, ma'am?
Email.
Okay. Yeah, I received it. Just sharing it.
Yeah, thank you.
Is it visible, ma'am?
Yes, I think so. It is visible to me.
It's visible.
So shall we start, ma'am?
Yeah.
So a very good afternoon to all of you, and sorry for this disturbance. I would like to start now. My topic today is about microbes, how they are important as biofertilizers, especially in agriculture, which is a huge sector in our India, we can say, and globally also, because we are dependent for our own food on agriculture. So we will be talking about its types, how microbes are used as biofertilizer, what are their types, what is their importance, and what are the benefits. So I'm Dr. Seema Sangwan. I welcome you all on my today's presentation. So next slide. So before starting this biofertilizer, the term biofertilizer, before starting about talking about this, we should know some basic facts about this. So please scroll on one by one. So first we will be knowing what is a biofertilizer, what are the fertilizer, we can say that fertilizer, these are the chemicals which are we are providing in our soil to fulfill the requirement of the plants, because as we need vitamins, minerals, everything, we need food for our growth and all. So the plants also, they need their macro and micronutrients which are available although in the soil from where they are taking this. But yes, when they start depleting, because there is a saturation point always comes now, which creates a depletion zone. After that, the soil or anything, they can't provide that input continuously. So to replace that thing, to increase the fertility or to increase the output of the plant or to increase the yield, fertilizers are usually used, which consist of any chemical composition that will provide directly or indirectly all the nutrients, macro and micronutrients to the soil. And that will be provided directly, indirectly to the plant then. So these macro and micronutrients, they consist of nitrogen, potash, and phosphorus. These are the macronutrients. And apart from that, many micronutrients are also there. So I think everybody knows about the Green Revolution, what happened at that time and what was the situation behind that. Before that Green Revolution, we were short of our food. And so Green Revolution, at the time of Green Revolution, many high yielding varieties were brought in India from Mexico, and we start growing them, those high yielding varieties. But there also comes the role of chemical fertilizer at that time, because chemical fertilizers were also used. Before that, nobody was using that chemicals के हमें कुछ पता ही नहीं था कि नाइट्रोजन कम है, फास्फोरस कम है या पोटाश कम है और इसको देने से क्या होगा या क्या नहीं होगा, ऐसा कुछ आइडिया था नहीं. लेकिन yes, our scientists, they did their best and they found out that हाई यील्डिंग वेराइटीज तो इस्तेमाल करेंगे ही, बट उसके साथ साथ केमिकल फर्टिलाइजर्स अगर डाले जाएं, उनका न्यूट्रिशन कंप्लीट किया जाए, तो आउटपुट विल बी like a boom. So it happened also, as we all know that how the Green Revolution changed the face of India in terms of food production. But yes, after 50 years now, this Green Revolution happened in 60s and 70s. But after 50 years now, we know that what happened due to these chemical fertilizers, what happened due to the continuation of using these chemical fertilizer. हमें बहुत सारे health issues हुए और बहुत सारा environment pollution हुआ, soil degradation हुआ, and yes, we posed a severe health hazard. So that is the main reason that we came to a conclusion that yes, we need some solution instead of these chemical fertilizers. India is the third largest producer and consumer of fertilizer in the world after China and USA. We are in terms of consumption, we are consuming 12% of the world population of N and P nutrients, especially अगर nitrogen or phosphorus nutrients के terms में बात करें, तो we are consuming 12%. And in terms of NPK nutrient, because there are some solution which are which comes in the form of a complete, we can say, package of nitrogen, phosphorus, and potash. Here we are consuming 12.6%, which is very huge. So we can see that how much chemicals we are consuming. So out of the total 329, we have 329 million hectare of India's geographical area, out of which 114 million hectare is under cultivation. And as India is growing and growing towards towards the most populous country, and how to meet the increasing food demand. So here comes a major challenge, and we need some solution. So to overcome this solution, how it can be done? There are two ways to overcome this challenge. First is to increase the food production, whatever the soil or whatever the area, cultivable area which is present with us, we have to increase, we have to do some technological work, or we have to do some magic so that the food production can be increased in the existing area. Or second solution is that we have to increase the area under cultivation, because अगर कल्टीवेशन एरिया बढ़ेगा तो कल्टीवेशन अपने आप बढ़ जाएगा, और अगर कल्टीवेशन एरिया हम नहीं बढ़ा सकते हैं, जो कि रियली पॉसिबल नहीं है, बिकॉज सबका ज्योग्राफिकल एरिया एक डिफाइंड है और उसको चेंज करना इट इज वेरी डिफिकल्ट. सो द ओनली ऑप्शन विद अस इज that we have to increase the production in the existing cultivation area. Here comes the role of biofertilizers, that instead of using the chemical fertilizer, which are, we know that they are highly dangerous, so we have to find some solution which can be used as fertilizer instead of these chemical fertilizer, so that the production and yield of the plant can be improved. Next. Please move the slide. So now we should know how, what are the reasons why crop productivity in India is decreasing. We are a country where we know that our 45 to 50% population is related to agriculture. They are working in agriculture. So why even then, why our crop productivity is decreasing in India especially? There are some causes for this especially. So first cause and the first most important thing, which the mistake we are doing, that there is an imbalanced and indiscriminate use of chemical fertilizer. Since Green Revolution, we can say Green Revolution start हुआ, it was okay for us because ठीक है, हमारे पास कम था food और एकदम से Green Revolution की वजह से boom हुआ और हमारे यहाँ yield बढ़ने लगा. But उसके साथ साथ हम continuously imbalanced और indiscriminate use करते रहे chemical fertilizers का, and due to which we are standing in a situation now where we know that how dangerous it is to use the chemical fertilizers. So that is why we are talking everywhere about organic farming or replacing the chemical fertilizer. So next question is that our plants, they need various macronutrients. So pata hi hai sabko ki yes, they need nitrogen, phosphorus, potash, but there are special micronutrients also, for example zinc, boron, sulfur, that play a very important role in their growth, in the growth of plant in agriculture, and they are not very easily available, and soil is very we can say it is so much degraded that these micronutrients, they are not present at all in our soil now, so that in the form especially which can be used by the plant. So this is also the reason. Next reason is that we have rain dependent agriculture. About 2/3 of our area is dependent upon rain. What does it mean? That we are totally dependent on the God's activity. We are dependent on the climate कि कब बारिश होगा तो ठीक है, अच्छा हो जाएगा, नहीं होगा तो हमारा बहुत सारा प्रोडक्शन खत्म हो जाता है, इसी वजह से उसमें डैमेज हो जाता है, या जितना होना चाहिए था उतना नहीं होता. तो that is the main reason also why our crop productivity in India is decreasing. So next is that inadequate irrigation facilities. We have irrigation facilities, yes, we know, but itna inadequate hai. That is why we nowadays it has been very much, we can say that it is very much facilitated in form of drip irrigation. We know that how drip irrigation is used to just to avoid the wastage of water. So, but in earlier times, and if we talk about the people which are living in village, they don't know about irrigation facilities and all, and at some areas are there in India where these irrigation facilities are not there at all, so they are totally dependent on the climate. That can be the reason also. And one important reason also that we don't even think about is that there is a continuous fragmentation of land and predominance of very small and marginal farmers. That is the reason that we are not, we have technology, but that cannot be applied just because there is a predominance of small and marginal farmers, and they are not able to adopt the technology. And that is the reason that crop productivity is not increased to that extent which it should be done till now. So I have told about this, that land holding pattern and predominance of marginal and small farmers. All these reasons, they totally in summation, we can say they affect the our crop productivity. So now we will be talking about biofertilizer, because we know we need some solution to replace the chemical fertilizer. Here the term comes the biofertilizer. What is biofertilizer? We know that chemical fertilizer means the fertilizer which is made of chemical, and biofertilizer means the fertilizer which is made up of some biological means. Especially biofertilizer, they are produced biologically, mainly using microorganisms. So here some living organisms are used, and they have some properties so that they can provide the plant chemical and fertilizer bio. Yes. Excuse me. Yes. If anybody is saying something. So biofertilizer, biofertilizers, they are produced biologically. That is why they are, the term has come biofertilizer, because here some living microbes are used in the form of fertilizer, which can provide the plant phosphorus, nutrient, and all the micronutrients also that we will be talking about in our next slides. So how important is the soil microflora? How it is important that it can be used as biofertilizer? So biofertilizers, they can be expected to reduce the use of chemicals and chemical fertilizer and pesticides. Yes, we know that. And this soil microflora, which is present in the soil, it has some, these microflora, they have been studied so much that we know that there is a group of organisms which we say that plant growth promoting microbes, we can name them. These microfloras, they have the ability to fix the nitrogen, to solubilize the phosphorus, to mobilize the phosphorus, and to hydrolyze the potash which is present in the soil, and to provide various micronutrients also as for example, zinc, boron, on sulfur to the plant, and the plant get these nutrients, and there is no need to give the chemicals. So here, and that is why the soil microflora, it becomes as much important in terms of biofertilizer. The microbes in biofertilizer, they restore the soil's natural nutrient cycle and build soil organic matter. They play an important role, that they are an important part of this soil organic matter, and they are present in the soil only. That is why they restore this natural cycle, which is already going on. They are restoring this. Next. Through the use of biofertilizer, healthy plants, they can be grown, and while enhancing the sustainability and health of the soil. We know that biofertilizer, they have very less, they are very, I don't think they have any significant harm to the plant. They are very, very useful. They are healthy. In the presence of these biofertilizer, healthy plants can be grown, and that will also be very helpful for the soil to increase the soil fertility also, which has been degraded due to continuous use of the chemical fertilizers. They are extremely advantageous in enriching soil fertility and fulfilling plant requirements by supplying the organic nutrients. As I can say that they are an important part of soil organic matter. So they are extremely advantageous, how to enrich, and they are enriching the soil and they are fulfilling the plant requirements. So biofertilizer, as I have already told, they do not contain any chemicals which is harmful to the living soil. And since their role as beneficial bacteria, we term them as plant growth promoting rhizobacteria. There are many studies, many papers which have been already published. It is a very well-known term, plant growth promoting rhizobacteria, because the bacteria which are present in the rhizosphere we will be talking about in a little detail in next slides, where we will be talking about what is rhizosphere, what is that place where these plant growth promoting rhizobacteria are present especially, and how we are isolating them, how we are mass producing them, and how we are checking them for their plant growth promoting activities. So next. Yes, concept of biofertilizer. As we know that this is not a new concept at all. Biofertilizer, it goes back to as early as 300 BC. When our ancestors, they talk about legume crops, they saw that these legume crops, for example, soybean, they have some nodules, they have some special structure in the roots. So the perspective of biofertilizer came from that point, because they were thinking that there is something which can be in these nodules which have the ability to fix the nitrogen. The first time they were talking about the rhizobium. Rhizobium is a bacteria which is present in these nodules, and it has the ability. With time, when it was studied, they found that it has the capability to fix the atmospheric nitrogen. So with time, many more researches were done, and it was found out that many bacteria, they were able to solubilize phosphorus also. There were some fungus that has the ability to mobilize the phosphorus. One is fixing the phosphorus, one is solubilizing the phosphorus, and one is mobilizing the phosphorus. So you can see how their activity and their communication, how they are acting in to carry out a particular we can say work. How I'm not getting that term कि कैसे वो एक साथ अपना communication करके एक ही nutrient को कैसे एक solubilize करता है, फिर एक mobilize करता है, and it finally reaches to the plant. So like this, potash solubilizers were also found out, and many more PGPR, plant growth promoting bacteria and fungus, they were found out. So the plant nutrition can be fulfilled easily. The role of biofertilizer assumes special significance due to increased cost of chemical fertilizers, especially, and their ill effects on soil health, as we all know that. Yes. So biofertilizer, we can say, and there is a term used that they are organic boost. Organic word comes from that because they are something living. They are some living organism which are providing the nutrition to the plant and which are increasing the fertility of the soil. That is why the word come organic boost. They are like an organic booster dose, we can say, a booster, which is pushing the plant to increase its growth, and which is pushing the plant to take up all the nutrients, and to increase its own health, and to increase its own yield. So the term biofertilizer, it refers to the preparation containing live microbes, which helps in enhancing the soil fertility, either by fixing nitrogen, by solubilizing phosphorus, by mobilizing phosphorus, or by providing any kind of, there are some microbes which are producing some plant hormones which have the ability to affect the plant growth directly. They are producing some we can say siderophores and indole acetic acid and extra many kind of hormones and many kinds of secondary metabolites, which affects the plant health and soil health also in direct or indirect manners. So like here comes the this concept about why they are organic boost. Next. So before starting about their types and all, we should know about rhizosphere. What is rhizosphere? Rhizosphere is a term used to the soil adhered to the root. The root, which is a plant root, and the soil which is adhered to that plant root. These soil is defined by the exudate, which are present in the root of the plant, and these root exudates, these are secreted in the surrounding environment, and that defined the microbiome of that area. We can say which type of fungus or which type of bacteria will be living there. That is totally different. So this zone, it is under the influence of the root. Then comes the term mycorrhiza. I have written these terms because they are highly confusing. Many times we see that mycorrhizosphere, what is this? Rhizosphere, what is the difference between these two? So rhizosphere is the term used for the soil which is directly under the zone, under the effect of the plant root. Mycorrhizosphere terms comes for the mycorrhiza. There are some, there is a special category of the organisms which are also, yes, they are biofertilizers. That is called mycorrhiza, and their spores are present in the we can say the roots and the soil, surrounding soil, and the zone of soil which comes under this influence of the root, which is already colonized by mycorrhiza, that is called mycorrhizosphere. These mycorrhiza, they have some hypha also. These are thread-like structure. They have their own flora. They have their own we can say microbes, fungus or bacteria, which are highly beneficial to the plants. So these hypha, the zone of soil which comes under this hypha, that is called hyposphere. So this is the plant we are watching, and here these are the roots. This zone which comes under directly comes under the zone under the influence of the root, that is called rhizosphere. When these plant roots, they are colonized by mycorrhiza, and then the adhered soil surrounding that, which is influenced by that root which is already colonized by mycorrhiza, that is called mycorrhizosphere. And there are some hypha also which are present in the soil. They are scattered everywhere, and there is some we can say microbiome, the fungus and bacteria which is adhered to this hypha, that is called hyposphere. So it is very clear from this picture that what is the difference between rhizosphere, mycorrhizosphere, and hyposphere. And these three zones are the most enriched zone, we can say, to isolate the plant growth promoting bacteria or any fungus. So these are the main center where we are, from where we are isolating, we are checking the their ability of microbes, whether it is a bacteria or fungus, whether they are able to do some plant growth promoting activity or not. Next. This is the list that shows types of biofertilizer which are very commonly present now. This is the category in which most of the work is usually done in terms of biofertilizer. So the broadest category is nitrogen fixing biofertilizers. These biofertilizers, they have been, we can divide them further also in free living form. Some are some bacteria have the ability to which are available in free, which are living in free soil. freely in soil. They are not associated with the root or with any other organism, but they have the ability to fix the atmospheric nitrogen. Atmospheric nitrogen, we know that it is available this is the largest gas which is present in the largest volume which is present in the environment, nitrogen. So it can be fixed by these bacteria. These bacteria have the ability to fix this nitrogen into available form that can be utilized by the plant. So this is the category of free living, where we can see that azotobacter, these are the names of some bacteria, Clostridium, Klebsiella, and Abina nostoc. They have the ability to fix this nitrogen. And these organisms, they are freely present. So, but beside this, there are some organisms which can only and only work in symbiotic association. Symbiotic, where there are two partners especially, and both the partners, they are getting benefits from each other. So rhizobium, for example, it is always present in legume plants, and in symbiotic association with the roots, they have the ability to fix the nitrogen which is present in the atmosphere, and that nitrogen is utilized by the plant. So plants have the ability to get these macronutrients. Fine. Beside, after that, this nitrogen fixing biofertilizer, we will come to the phosphate solubilizing biofertilizer. We know that after nitrogen, phosphorus is the main component which is needed by the plant for their growth. And here also we have a category of bacteria and fungi who have the ability to solubilize the phosphorus, which is already usually present. Phosphorus is usually present in the soil, but it is not in the available form. It can't be utilized by the plant. There is very little percentage which can be utilized by the plant directly. Plant roots, they can, we can say, they can take up that phosphorus, but yes, after that, a depletion zone is, they are not able to take up that phosphorus. So these bacteria and fungi, they have the ability to solubilize the phosphorus which is present in the plant, and it is converted into a form which can be now soluble form, and which can be utilized easily by the plant. So these bacteria and fungi, for example, examples are for bacteria, Bacillus megaterium, Bacillus subtilis. Bacillus is the main species here, and Pseudomonas. Bacillus and Pseudomonas, they are the main category of, they are the main genus of the bacteria which comes under plant growth promoting bacteria. So some fungus have also have the ability to convert to solubilize the phosphorus, that is Penicillium aspergillus. After that comes the phosphate mobilizing biofertilizer. Here the most common biofertilizer, we can say, most common organism which is a fungus, arbuscular mycorrhiza fungi, that have the ability to to mobilize the phosphorus from the soil to the plant root. I will be talking about this in little detail about arbuscular mycorrhiza, because this is my area of research nowadays, and I just want to share how it works. So after that, there are some other mycorrhiza also which have the ability to mobilize, but the most important is arbuscular mycorrhiza fungi. So after P mobilizers, we come to some micronutrients, some organisms, some bacteria, especially Bacillus and Pseudomonas. They have the ability to solubilize zinc also, to solubilize silicate also, which is present in the soil. And these micronutrients then are utilized by the plant. Next. So in this picture, we can see that how they look like. In this slide, I have made especially just to see how the rhizobium looks like in the Petri plate, how azotobacter they look like, how the PSB. Here you can see that these are the clear zone. PSB means phosphate solubilizer. These spots, in white spots in between, they are the organism, and the clear zone surrounding them, it is showing that they are solubilizing the phosphorus which is present in this media, and after solubilizing they are creating this clear zones. Fine. So these are phosphate solubilizing bacteria. Like this activity they are doing in soil also, and that is why the plant is able to take up that phosphorus. So blue-green algae, how they are looking like, and these are the arbuscular mycorrhiza that we'll be talking in a little detail, and these are the azospirillum, which are the nitrogen fixers. Yes, next. This is the microscopic view of my own work that I have isolated some of the PGPRs, some of the plant growth promoting bacteria, and how does they look like in under microscope. When we saw them, they have different structures. Some are rod shaped, some are cocci shaped, some are in bunches, and some are in diplococci. They are present in pairs. Some are large in size. Rods are large, and in some and they are very small. So this is just a microscopic view how these different bacteria they look like. Yes, next. So first we will be talking about nitrogen fixing biofertilizer. The main organism under this category comes the rhizobium, which is a symbiotic, which always works in symbiotic association with the legumes. This is a soil habitat, this is a soil bacteria which is able to colonize the legume roots. It forms nodules there, and it fixes the atmospheric elemental nitrogen symbiotically into plant usable form. And it has the ability to fix the 50 to 100 kg per hectare per year of nitrogen. And that is why it is very much important in concern when we want that. Yes, our plant need nitrogen. Here, their role, the role of rhizobium, especially in legume plants, it is very important. And that is why it's important for the growth and for the production of legumes and oilseeds especially. Next. Now, cyanobacteria, they are also nitrogen fixers, but the main difference between the rhizobium and the cyanobacteria is that they are both free living. They can free live also, and they can act symbiotically also with they are specially used. These cyanobacteria, they are specially used in rice, and they the benefits due to alkalization, it could be stored to the extent of 20 to 30 kilogram per hectare. And these cyanobacteria, they have the ability to produce some growth promoting substances also. For example, they are producing vitamin B12, and they are also producer of especially indole acetic acid, which is a plant growth hormone that directly affects the root architecture of the plant. So they improves the soil aeration, they increase the water holding capacity of the soil, and all these biofertilizer, they finally add up. This is this is a common statement that they add the biomass when they decompose after life cycle. Sorry. So next slide. Now, azospirillum, they have the ability, they are also nitrogen fixers, and they can fix 20 to 40 kg per hectare of nitrogen. They can provide this much nitrogen when they fix, and they can proliferate under both anaerobic and aerobic conditions. They can also produce indole acetic acid, and they can also produce some they can also produce some compounds which are highly active against pathogens. So they make the plant where they are present, and if we are using azospirillum, they make the plant resistant to these pathogens. And they also increase the ability of the plant to tolerate the drought. Means when water is less, these azospirillum, they works very well. They fix nitrogen also, and they increase the ability of the plant to tolerate the drought or when water is less. So these are some additional benefits. Next. After that, azolla. Azolla is a free floating water fern which is used as a biofertilizer. Especially it is used for wetland rice, and they are also nitrogen fixers. They fixes atmospheric nitrogen in association with nitrogen fixing blue-green algae, that is Anabaena azolla. So they work in symbiotic association, and they also contribute 40 to 60 kilogram nitrogen per hectare per rice crop. So we can say that they can, when we are using some chemical instead of these, and when we start using these azospirillum azolla, so we can anybody can think that the question comes in our mind that how much chemical they can replace. मतलब हमें कितना कम कर देना चाहिए अपना chemical, क्या ये completely उसको हटा के use किया जा सकता है? तो I want to show you guys that all these biofertilizer till now, most of the biofertilizer, they can replace the 30 to 35% and up to 40% of the chemicals, especially if we talk about nitrogen fertilizers. So they have the ability to replace chemicals up to 40%. So we can use these biofertilizer and it will be very helpful. So next, after comes after Azolla, Azotobacter. All these are the example of nitrogen fixers only. So Azotobacter also, it is a free living bacteria mostly found in neutral to alkaline soils, and it has the ability to fix nitrogen as all are doing, and it also produces abundant slime which helps in soil aggregation, and finally it plays an important role in increasing the water holding capacity of the soil. And they also produce indole acetic acid and gibberellins, which are the plant hormones. So these are some additional on all these bacteria which are used as biofertilizer. They have some important trait, the main trait, and beside that they have several other traits also which are very much beneficial. That is why the term plant growth promoting bacteria is used for all these organisms. So next, after nitrogen fixers come phosphate solubilizing bacteria. So phosphate solubilizing biofertilizer, in as I have told a little bit about that phosphate is present in soil, but is not available for the plant. Plant is not able to use that because that is in the insoluble form, that is present in insoluble form. So when we use phosphate solubilizing biofertilizer, when we are adding some microbes which have the abilities to solubilize that phosphate, so that is converted into a form which can be easily utilized by the plant, which can be easily taken up by the plant. And that is why these phosphate solubilizing biofertilizer, they play important role to fulfill the requirement of the plants, especially the phosphorus. So the genera which plays an important role here as phosphate solubilizer biofertilizers are Pseudomonas, Bacillus, and Rhizobium especially. They are the most powerful among these bacteria. Yes. After that phosphate solubilizer comes the phosphate mobilizer. These two are totally different. Phosphate solubilizer means they are just converting one form into another, insoluble form into soluble form. But yes, there are a special category of organism, microorganisms, especially the fungus, which have the ability now to mobilize. These phosphate mobilizing biofertilizer, they also live in symbiotic association, and these have the ability to make a network, a channel between the soil where the phosphate is present in soluble form and the plant root. So this channel, it works like a we can say bridge between these two hosts, and through this bridge all the phosphate is easily mobilized. From the depletion, beyond depletion zone also, this phosphate is mobilized into the plant, and plant is very much able to grow and to fulfill its requirements. So yes, here our work comes as phosphate mobilizing biofertilizer. Next. So some silicate and zinc solubilizer biofertilizer are also there, which have the ability, these Bacillus and Pseudomonas species, they have the ability to provide to degrade the, we can say that to provide the zinc and silicate to the plant, so that these micronutrients, they can also be easily taken up by the plant, which is usually not very much possible. And many of the chemical fertilizer, they don't give, they only provide NPK especially, and they are not very much, we can say there are not many options in chemical fertilizer to fulfill the requirement of the plant, which is in the form of micronutrients. So here the role of biofertilizer is very important, and these have the ability, some of the genera, bacterial genera, they have the ability to provide silicate and zinc to the plant. So this micronutrient requirement can also be fulfilled through this way. So plant growth promoting rhizobacteria, that this term I have written just to any bacteria, any fungus which have the capability to increase the growth of the plant in any form, whether they are phosphate solubilizer, whether they are nitrogen fixers, or phosphate mobilizer, or zinc provider, or silicate provider, or potash solubilizer. All these bacteria comes under a broad category, that is plant growth promoting rhizobacteria, because beside this, they are also producing various plant hormones, various secondary metabolites, which are very helpful for the plant growth, and they which make the plant very much tolerant to the extreme condition. For example, whether it is salinity, whether it is drought or any extreme condition, these organisms, they make the plant very much tolerant to these conditions. So that is why the terms is very commonly used, that is plant growth promoting rhizobacteria. So I will talk about mycorrhiza especially because it is we can say that among all the biofertilizer, they are very much known. There are many studies already have been done, but mycorrhiza especially, it is a it is a category of biofertilizer which has come under the fertilizer control order in 2008 only, or 2008 or 9, I'm not sure, but it is very much recent category which have been included in the biofertilizer. Or what is mycorrhiza? So mycorrhiza, they are the fungus root. We can say that the word mycorrhiza, that means fungus root. They are like a symbiotic association which is present with the plant. This fungus, mycorrhiza, arbuscular mycorrhiza fungi, this is a fungus which lives in symbiotic association with the होस्ट प्लांट. कोई भी प्लांट है उसकी रूट्स के साथ इसका एसोसिएशन होता है, जिसमें ये उसके रूट के अंदर जाके, मतलब ये प्लांट, ये माइकोराइजा जो हम कह रहे हैं वो इन द फॉर्म ऑफ स्पोर्स और एनी अदर इन्फेक्टिव प्रोपेग्यूल, दे आर प्रेजेंट इन सॉइल, and when the host comes, they start colonizing the soil, colonizing the plant root, where they are making a channel. Through this channel, all the nutrients, all the we can say water, especially all the nutrients, especially the phosphorus, they are very easily go towards the plant. So plant is able to easily take up these nutrients and water from its surrounding. That is why mycorrhiza, it is very much popular nowadays in the form of biofertilizer. Next. These are of two types, especially ectomycorrhiza and endomycorrhiza. Ectomycorrhiza, they are just able to make a channel with the upper surface of the plant root. They can't enter into the root, but they are just making a form of net with the soil and plant root. So endomycorrhiza, they are making this association inside the root. So they have these endomycorrhiza, they have the ability to enter inside the root of the plant, and they are just forming a network, a web channel surrounding the plant root, that between the soil and the plant root. That is why all the nutrients, especially, and water, it can be easily transferred to the plant. Next. So we are talking about arbuscular mycorrhizae only. They are also called nature's own biofertilizer, because 80 to 85% of the plant, if we check their root just now, if we randomly check the roots of the plants, they will be already having this biofertilizer in their roots. So that is why this, especially the arbuscular mycorrhiza fungi, it is present in 85% of the plant roots already. So they are called nature's own biofertilizer, which was already present. So they have the ability to improve the soil structure, they increase the plant health, and also they decrease the stress caused by pathogenic bacteria, fungi, and other pollution due to heavy metals. They have several infective propagules through which they work, that is spores, hypha, vesicles, and arbuscules. Next. Spores, I will be taking very less time, because this is, I think it may not be very much useful for you, but you should know about the spore, that yes, they are the spores which are present in the soil, and they are the swollen structure. They are very much larger. We can see them through microscope, using microscope, not through the naked eye. And these spores, they are the, we can say, they germinate themselves, and then they start moving towards the root, and where they are forming this network, where this is the starting point, kind of. Next. After spores come the hypha. I have talked about hyphosphere also. So these hypha, they are thread-like structure. They form a mycelium of this fungus, and this hypha only, it forms in clubbing. They form this network between the plant root and the surrounding soil, through which all the water, all the nutrients, they are easily going to the plant. So this hypha, these thread-like structure, they play a very important role. Next. These microscopic pictures, this is my work only, where I have in this first picture, you can see the hypha, how they look like. And these picture I have taken under compound microscope. And here you can see the spore, which is present inside the root. This is the root. I have taken the root picture where the infection due to where the arbuscular mycorrhiza is colonizing this root, and it how does it look like inside the root. This is the picture shows this, that how does they look like inside the root. These are the individual spores that we have taken out, and we have studied them, how the this is the germination tube they have, these spores, and how does they look like. And there are many, around 150 to 200 species only which have been studied, which have been found out in which have been classified till now in under arbuscular mycorrhiza fungi. So there is a lot of work to be done in this area. So next, these pictures also show the plant roots. How does they look like when these arbuscular mycorrhiza, they colonize the root, and like this, they are present inside the root in this form, and they will be forming a network with the surrounding soil, through which all the nutrients, and they are just the mobilizer, especially through which all the nutrients and water will pass very easily. We know ना that ठीक है, हमारे पास दो अलग अलग पॉइंट्स हैं, एक यहाँ पे है, ए है और एक बी है, लेकिन इनके बीच में जब एक नेटवर्क हो, जब एक चैनल हो, तो उसके थ्रू कोई भी चीज इजिली ए टू बी जा सकती है और बी टू ए आ सकती है. दैट इज द मेन बायोफर्टिलाइजर. दिस बायोफर्टिलाइजर इज प्लेइंग दिस रोल to mobilize the things from one point to another, that is from soil to the plant. Next. So root colonization by our Pusa mycorrhiza, we in our IRI, Indian Agricultural Research Institute, in Division of Microbiology, we are producing this Pusa mycorrhiza, and this plant shows very clearly what is the difference by using mycorrhiza or as a biofertilizer. This plant, it does not have anything. It is without inoculation of mycorrhiza, and this is after inoculation of mycorrhiza. So you can see the difference between these two, how biofertilizer, they can really do this magic. Yes. Next. So benefits of arbuscular mycorrhiza, I have already told that they are the channels through which all the nutrients that goes very easily, because they increase the rootlet size and longevity, especially the roots are very dense, the volume of the roots is increased, the surface area of roots are increased. That is why the nutrients are very easily taken up by the plant, and yes, they help in water uptake also, and overall improve the growth rate of the plant. Next. Now, production of biofertilizer, we know that how the production will be done. That I have made I can say that in next slides, please do the forward. Go to the next slide. Next. I have made a flow chart where we can easily see that how they are produced, how this biofertilizers is produced, how we start it in the lab. So we prepare, especially we prepare appropriate media for a specific kind of bacterial inoculant. We know that we will be working on phosphate solubilizer or nitrogen fixer. So when we for example, if we found out that we have to isolate some phosphate solubilizer, so there will be a appropriate media related to that, and we will be preparing that media, and through which the bacterial strains, they will from the soil, they will be allowed to grow in that. For example, if we are taking phosphate solubilizer, if we are isolating them, so we will be taking a Picovaskia media is used for phosphate solubilizer. So we will be taking soil samples, and we will be doing that after dilution. If we plate them on the particular media, then some of the organisms who have the capability to solubilize the phosphorus, they will only grow and they will form a zone after 2 or 3 days. And we can pick those colonies and we can do our further research on those colonies, and they can we can classify after that. If they are producing some other PGPR traits also, we can classify them as a plant growth promoting bacteria. So there are some We know that every bacteria, they have their particular temperature of growth, and especially that log phase, how much they will be up to which state they will form the 10 ki. We know that in biofertilizer we are maintaining this population of 10 ki power 8 to 9 cells per ml. So to make it, every organism has its own time to go to the log phase of this culture. So we are making that. Our motto is to make the final population load of 10 ki power 8 to 9 cells per ml or per gram of any carrier or any media which we are using, so as a biofertilizer. So carrier materials are used specially because just to increase the we can say the longevity or shelf life of the organism, we are using carrier material also, and to increase their activity also, so that they will be active, and when we apply them in the soil, they will be producing their traits, and through which they will be helping the plant in growth. So there are many carrier materials are used during production. For example, peat, soil, lignite, charcoal, pressmud. We are using charcoal in our department, and farmyard manure can also be used. And in case of mycorrhiza, we are using soil only as a soil. These are the soil-based culture also. So yes, we can use any kind of carrier material, but it has some ideal characteristic that it should be cheaper and it should be locally available. It has some organic matter content, because that will be very helpful, because it will increase the organic matter content of the soil, that will directly increase the soil fertility. There should be no toxic chemical, and water holding capacity should be more, so that the water uptake is increased for the plant, and they should be easy to process. So like this, carrier materials are chosen. Next. So now comes the application method. We know that we have some biofertilizer. Yes, they are helping the plant, but how to treat? Finally, we have made the biofertilizer, or we are getting any biofertilizer from the market also. How to treat? There are 3 special treatments which are done. Especially one is 3 ways of treating the of applying these biofertilizer. The most important and most common is seed treatment. If we treat the seeds in any way, कोई भी तरीका आप इस्तेमाल करिए, कैसे भी, अगर आप सीड को ट्रीट कर देते हैं सीड की कोटिंग करके या किसी और मेथड से, कैसे भी, तो जैसे ही सीड जब अपनी ग्रोथ जर्मिनेशन स्टार्ट करता है, वहीं से एक्टिविटी ऑफ द बायोफर्टिलाइजर स्टार्ट्स. उनका जो भी ट्रेट है, जो भी एक्टिविटी है, वो नाइट्रोजन फिक्स कर रहे हैं या फिर फॉस्फेट सोल्युबलाइज कर रहे हैं या कुछ भी कर रहे हैं, तो वो वहीं से उनके फर्स्ट डे से ही स्टार्ट हो जाता है. दैट इज व्हाई इट इज द बेस्ट मेथड. एंड सम ऑफ द ऑर्गेनिज्म, दे वर्क्स वेरी वेल व्हेन दे आर वेरी मच नियर टू द सीड, बिकॉज जैसे जैसे प्लांट होगा और जितना वो उसके क्लोज होंगे, वो उतना ही एक्टिविटी अपनी करेंगे, और दैट बेनिफिट विल डायरेक्टली गो टू द प्लांट. दैट इज व्हाई seed treatment is the most important way to treat. But yes, in some cases, for example, in case of rice, when transplantation is done, here comes the method where we dip the roots in the solution of biofertilizers. So root dipping method is also a method, but it is used in some of the crops, especially rice. And yes, the third method is soil application, where we treat the soil. Before sowing, we treat all the soil with the biofertilizer. We used to mix them in the upper layer of the soil, so that when we will be doing the sowing, after that the effect will automatically it will go to the plant. So the plant will get the benefit of these biofertilizer, and it will be helpful for its own growth. Yes, these are the 3 ways: seed treatment, root dipping, and soil application. But yes, seed treatment is the most common method which is adopted for all the inoculants. So seed treatment with Rhizobium, Azotobacter, Azospirillum, and phosphate solubilizer microbes is usually done. There are no side effects, and it is a very it can be done with any of the, और मतलब एक बैक्टीरिया है या एक से ज्यादा आपका कंसोर्शिया, ऐसा कंसोर्शिया है जिसमें एक से ज्यादा आपके पास माइक्रोब्स हैं, तो किसी भी मेथड को किसी भी ऑर्गेनिज्म के लिए सीड ट्रीटमेंट कैन बी यूज्ड. नेक्स्ट slide. Root dipping is usually done for the paddy crop and vegetable plants also, where transplantation is usually done, or azospirillum, which is a free living bacteria. तो इसके लिए this is the best method कि आप उसका कल्चर रेडी करें और उसको रूट में जैसे वो ट्रांसप्लांटेशन के लिए रेडी हो उससे पहले आप उसको रूट्स में को उसमें डिप करें और फिर उसको ट्रांसप्लांट कर दें. So root dipping is the second method. Now next. And third one, as I have already told, soil application, where the organism is usually mixed with the upper layer of the soil. The quantity which has to be applied with the according to the area that usually differs with the inoculant, from where we are getting that. Its strength के कितना strength है, कैसा है. कई बार कम strength का आपके पास biofertilizer हो सकता है, तो उसके लिए आपको ज्यादा amount use करना पड़ेगा. And if the it is very much enriched, तो आपको कम use करना पड़ेगा. It totally depends on the kind of biofertilizer and the source from where you are getting. So according to that, you can finalize कि कितना आपको use करना होगा per acre और per hectare. And yes, soil application में आप ऊपर वाला soil में आप इसको मिला दीजिए रोज बना के या कैसे भी, और उसको फिर leveling कर दीजिए. After that you can sow your seeds, so it will produce its effect. Next. There are some precautions also related to biofertilizer that we already always they all should be in our mind when we use the biofertilizer. That first and most common thing is that these biofertilizer packets, they should never be stored in hot area or under direct heat. They should always be placed in cool and dry place, because these are the living organism. And yes, you know that if we can't tolerate sunlight or a heat up to after a point, so these bacteria, they also have a minimum and maximum range of the temperature where they grow. So yes, according to that, they always need to store in cool and dry place. And we should always use right combination of biofertilizer, because it is very important. They should not be competition. There should not be competition. If it is a consortia, or if we are using two different biofertilizer, one is phosphate mobilizer or another is nitrogen fixer, there should not be a competition between them. They should be compatible. And some are very crop specific, so you need to be very specific where you are using, which biofertilizer you are using, and for which crop you are using. For example, some are very much general. For example, arbuscular mycorrhiza fungi, it is already present with 85% of the plants, so it is very general. It can be used with most of the plants. But rhizobium, it is very crop specific. It can be used in legumes and oil seeds only. So that you need to keep in your mind how to use and in which crop you are using. Do not mix them with chemicals, because they are living organism, and in chemicals there can be any pesticide which can affect the growth of these organisms, or which can inhibit which can kill the bacteria, or which can kill the fungi which we are using as biofertilizer. So we should not mix them with chemicals. And we should check कि एक्सपायरी कितना डेट है उससे पहले आपको एक स्पेसिफिक क्रॉप के लिए, और अकॉर्डिंग टू द रिकमेंडेड मेथड विच इज गिवन ऑलवेज ऑन द पैकेट, सो दैट यू नीड टू कीप इन माइंड कि कैसे यूज करना है, कौन सी डेट से पहले आपको यूज करना है और किस स्पेसिफिक क्रॉप के लिए यूज करना है. नेक्स्ट. So here are some advantages in we can say that after listening this why biofertilizer are what are the advantages, why they are useful. First and most common advantage is that they are renewable source of nutrients. Chemicals की तरह नहीं है जो कि एक दिन नॉन रिन्यूएबल है और एक दिन वो खत्म हो जाएंगे तो उसके बाद हम क्या करेंगे. तो दे आर रिन्यूएबल सोर्स ऑफ न्यूट्रिएंट्स. दे सस्टेन द सॉइल हेल्थ. दे डायरेक्टली अफेक्ट द सॉइल फर्टिलिटी. दैट इज व्हाई दे आर वेरी मच इंपॉर्टेंट फॉर आवर सॉइल हेल्थ आल्सो, बिकॉज आवर सॉइल हेल्थ इज डिग्रेडिंग day by day due to the continuous use of these chemicals and inappropriate use of these chemicals. And that is why the biofertilizers are very important. They supplement chemical fertilizers. They replace 25 to 30% of chemical fertilizer. This term, this line I want to specify, because we have this many times we think that yes, अगर biofertilizer दे दिया तो हमने पूरा का पूरा chemical उसको हटा देंगे तो equal आना चाहिए. That will never not be the case. Around 5 to 6 years it will take the soil to get adapt, which is already adapted with the chemical fertilizer. अगर हम एक चीज से adapt पहले ही हैं, हमें पता है कि बहुत easily हमें chemical fertilizer हमारे अंदर डाला और हमने grow करना start कर दिया. But जब chemical को बंद किया और उसकी जगह कुछ और शुरू किया, तो वो acclimatize होने में बहुत time लगता है, 5 to 6 years usually. This is the minimum period. That is why we say that in initial period of time, we should always replace a percentage of these chemical fertilizer. It can be easily done up to 30%, 35%, in some cases 35% also. And after that, धीरे धीरे आप इसको totally replace कर सकते हैं. So it will increase the grain yield by 10 to 40%. It will, they will also decompose plant residue which is present in the soil, and that directly stabilize the C/N ratio of the soil, which is very important to maintain the soil fertility. They improve the texture, structure, and water holding capacity of the soil. And there is no adverse effect on the plant growth and soil fertility by using these biofertilizers. So next. They stimulate plant growth by secreting various growth hormones. As I have already told that some of the biofertilizer, they have the ability to produce indole acetic acid, cytokinins or gibberellins, and they have also have the ability to produce some antipathogenic substances or antibiotics, so that they can kill the pathogens surrounding them, and they make the plant sustain these diseases. Matlab they can fight against disease, and they help the plant to fight against diseases, and they have the ability to solubilize and mobilize the nutrients which are present in the surrounding environment. They are eco-friendly because there is no use of chemicals, and they are non-pollutant because they are not getting we can say they are not getting accumulate in the soil, and they are very cost effective in comparison to the chemicals. Next. There are some disadvantages also related to these, which is very nominal kind of, but yes, they have some disadvantage when we compare them with the chemicals. These biofertilizer, they especially require special care for long-term storage because they are the live organisms. लिविंग कोई भी चीज होगी तो उसको थोड़ा एक्स्ट्रा केयर का जरूरत हमेशा होगा, और केमिकल्स की तरह जो कि एक नॉन लिविंग थिंग है जिसको आप कैसे भी रख सकते हो, कैसे भी कर सकते हो, बट यस हियर they are living organism. That is why this needs special care. They must be used before their expiry date, and if other microbes they contaminate the carrier medium, or if growers use the wrong strain, they are not effective, because here we are using microbes, so the culture needs to be very pure, whatever we are using. And if it is a particular organism, yes, it is doing some particular work, it is fixing the nitrogen, and if it gets contaminated with which can be easily done, मतलब contamination बहुत easily हो सकता है, तो अगर कोई wrong strain आ जाए तो वो जब आप डालोगे आपको पता भी नहीं चलेगा कि वो इफेक्टिव क्यों नहीं है, वो इसलिए नहीं है क्योंकि वो कंटामिनेट हो चुका है. दैट इज व्हाई यहाँ पे बहुत एक्स्ट्रा केयर का जरूरत होता है. Biofertilizer, they lose their effectiveness if the soil is too hot or too dry. Yes, extreme environments may, these are the live organisms, so they have their own range of growth, their on range of temperature range जहां वो grow करते हैं up to their maximum extent. And if it is used in some extreme soil which is very too hot or too dry, their effectivity usually loses. Next. These are just pics to show our products which are already available, which we are producing some many biofertilizer in our Indian Agricultural Research Institute. So some of the nitrogen fixers, potash solubilizer, phosphate solubilizer, iron providing organisms. All these are the packets of biofertilizers which are present in our sale counter in Division of Microbiology, IRI. Next. These are carrier-based, and these are some liquid cultures also. We have Pusa Bio Zinc, which provide the zinc, and Pusa Bio Potash. And these liquids, they contain specific microbes, specific bacteria, which are doing special, they have special characteristic. For example, Pusa Biophos, it is providing the phosphate to the plant. If we we can say, if we are giving the plant, they are providing the phosphorus. Like this azotobacter, which is fixing the nitrogen, and Pusa Sampung, that is NPK, that provides nitrogen, phosphorus, and potash, all the three macronutrient to the plant. Next. This is Pusa mycorrhiza, which I have told you guys about this mycorrhiza biofertilizer. It is a very, we can say, advanced kind of biofertilizer, which is very helpful for the plant. Next. Biofertilizer, they have a great role in increasing the crop production. Just give me a second, please. Just. Sorry for the interruption. Yes, these biofertilizer, they have a great role in increasing the crop production. They provide different growth promoting hormones and phytohormones to the plant, as I have already told you. And I have explained everything related to these biofertilizer. And the most important thing related to them, which makes them very much superior to the chemical fertilizer, is that they do not leave any residual effect like chemical fertilizer. That is why. Next. That is why these biofertilizer, they are the best option nowadays in sustainable agriculture. So to make our agriculture sustainable and to increase the productivity by decreasing the use of chemical fertilizer, there is only one solution, that is biofertilizer. Thank you. And now, please, if anybody have any question, you guys can ask me. I tried my best, but.
Thank you, Dr. Seema. If participants have any question, please interact. Dr. Seema, we have received one question that did you work on the indirect mechanism that were involved with microbial species that you were working with, the like production of the antibiotic or hydrolytic enzymes?
Yes, sir. I am working with mycorrhiza that I have told about in detail. Or here, the main work of mycorrhiza is to mobilize the phosphorus. That is, it provides a plant the phosphorus. But the indirect effects, that how it is promoting the growth of the plant, that is, it also produces some antibiotics. It is producing some plant hormones also. It produces indole acetic acid especially, which directly affect the plant root and increase the surface area and volume of the plant root, and that makes the plant more powerful in taking up all the nutrients and water from the soil. So these are the indirect ways.
Thank you, Dr. Seema. Yukta Tomar, you might get your answer. Any other students or any participant you would like to ask, you can drop in chat message.
Can I ask a question?
Yeah, sure, madam. You can ask.
Dr. Seema, it was a wonderful lecture and very simple way you have talked about. I just want to know, is there any, you are doing some awareness program for the farmers also?
Ma'am,
Because,
yes, we used to do, many times we are doing that, but it is not done through our division directly. It is done through extension division. There is extension division in our Indian Agricultural Research Institute, and their farmers are invited from all over India, from Rajasthan and Madhya Pradesh, all over India, they are coming. And they used to come, and there we are providing information related to compost and related to biofertilizer, everything.
thank you. Do your department also offer some internship program for the students like in summer? Can they come and
Yes, ma'am. In our institute, they are providing some options for the dissertation of MSc students. They used to, there are some channels through which students can come for 4 months or 5 months work, and they can complete their dissertation there. And there is some formal channel. I don't know about that because they
Is
can't
there any-
directly come to us.
Okay, thank you.
Yes.
Thank you, Dr. Seema. We have received one more question from the Ritu Dagar. Ma'am, why crop need time for adjustment with biofertilizers when they are also providing the same nutrient they are receiving from the chemical fertilizers?
Yes, they are receiving from chemical fertilizer, but we need to replace these chemical fertilizer. We can't eat them now. हमने बहुत खा लिया है, 50-55 साल से खा रहे हैं, और केमिकल्स की वजह से आजकल कैंसर के केसेस इतने ज्यादा हैं. एवरीबडी कैन सी के फार्मर्स में और कितने हेल्थ इफेक्ट्स इसकी वजह से बढ़ रहे हैं. दैट इज व्हाई द वर्ड ऑर्गेनिक फार्मिंग इज इन सो मच फैशन. कि सबको पता है कि ऑर्गेनिक फार्मिंग जो जिसमें केमिकल्स ना डाले जाएं, जो विदाउट केमिकल फ्री हो टोटली, वही हम खाएं, सो दैट हम अपनी अगली जनरेशंस तक भी हेल्दी, मतलब हेल्थ उसमें हेल्दी वे में जाएं. तो दैट इज द रीजन वी नीड टू रिप्लेस, वी हैव टू रिप्लेस द केमिकल्स. एंड yes, our soil is already acclimatized with these chemicals because we are using them since ages. And now if we are replacing them and we are doing something else, we are giving the plant something else, there they need the time to acclimatize, and that is the reason that biofertilizer they need, they take more time in comparison to chemical fertilizer.
Thank
I
you,
think
Dr.
I'm
Seema.
clear.
Yeah. One more question we have received from one participants,
Yes.
that is Rimi. She asked that, ma'am, how your organization that microorganisms are extracted for further process, or they are converted into the powder from one on any other medium?
Remain, there is a technical, we can say there is a technical lab work that is done in labs. We are first isolating a particular organism from the soil using special media. जिस भी ट्रेट के लिए हमें काम करना है, उसी ट्रेट के अकॉर्डिंग हम मीडिया चूज करते हैं और उस ऑर्गेनिज्म को टारगेट करते हैं. जब हमें लगता है कि हाँ, इसमें एक पर्टिकुलर ट्रेट हमें दिखाई दे रहा है जो इसमें हम इंटरेस्टेड हैं, फिर हम उसको स्टडी करते हैं फॉर फर्दर ऑल अदर ट्रेट्स. मतलब जो अगर ये प्लांट ग्रोथ प्रमोटर कैसे है, ये कोई इंडोल एसिटिक एसिड प्रोड्यूस कर रहा है या कोई और प्लांट हार्मोन प्रोड्यूस कर रहा है, और या फिर ये नाइट्रोजन भी फिक्स कर रहा है. बहुत सारे ऑर्गेनिज्म ऐसे हैं जो नाइट्रोजन भी फिक्स करते हैं, जो फॉस्फेट भी सोल्युबलाइज करते हैं, और जो प्लांट हार्मोन्स भी प्रोड्यूस करते हैं. ये सब देखने के बाद जब हमें पता चलता है, फिर हम उसको स्केल अप करते हैं यूजिंग, पहले छोटे फ्लास्क में लैब में ग्रो करेंगे और उसके बाद उसको बिग फर्मेंटर्स यूज करके उसको स्केल अप करेंगे. उसके बाद उसको जो भी ऑर्गेनिज्म हमारे पास आएगा उसको एक पर्टिकुलर कैरियर मटेरियल के साथ मिक्स करके उसको पैकेजिंग किया जाता है पर्टिकुलर तरीके से, एंड दैट इज हाउ इट कम्स इनटू द मार्केट.
Thank you, Dr. Seema. Very nice reply. And Rimi, I think you're satisfied with the answer. And Dr. Seema, we have another question from Priyanka Sharma. She asked that could rhizobium be slowly used as a biofertilizer, or does it need to be consorted?
Yes, it can be used. It depends on the requirement now of the plant. अगर कोई आप ऐसा प्लांट है, स्पेशली लेग्यूम के साथ ही आप राइजोबियम एक तो यूज कर सकते हो, रेग्यूम या कुछ पर्टिकुलर ऑयल सीड प्लांट्स हैं, उन्हीं के साथ आप राइजोबियम यूज कर सकते हो, वो किसी और के साथ नहीं यूज कर सकते. और यस, उनको कंसोर्शिया कंसोर्टेड फॉर्म में यूज किया जा सकता है इफ द प्लांट नीड्स. अगर उसको सिर्फ नाइट्रोजन का जरूरत है, नाइट्रोजन कम है और उसके पास फास्फोरस इनफ है और अदर माइक्रोन्यूट्रिएंट्स भी हैं, तो वहां आपको शायद जरूरत नहीं पड़ेगी राइजोबियम के साथ कुछ भी डालने की. बट यस, अगर नाइट्रोजन के अलावा वहां पे फॉस्फेट भी नीडेड है, तो राइजोबियम को आप कंसोर्टेड कर सकते हो विद सम फॉस्फेट सोल्युबलाइजिंग माइक्रोब्स. तो उसको आप ऐसे भी अप्लाई कर सकते हो, सिंगली भी अप्लाई कर सकते हो. इट टोटली डिपेंड्स ऑन द need of the plant.
Thank you, Dr. Seema, for a wonderful reply. We have another question from Manasa that she asked, ma'am, is there any training program for BSc student for production of the biofertilizer in IRI Pusha?
Mantasha. Actually, directly, you guys, we can't train them in departments. There is a particular department there, extension division, through which you can apply. And yes, we are giving these trainings, production of biofertilizer. We used to tell them all other things also, means how to make compost, vermicompost, fertilizer, everything, how to make a farmyard manure, everything is told. But yes, there is a specific channel through which you can apply and come.
Thank you, Dr. Seema. Any other question? If any participant want to interact with our speaker, you can interact, you can ask, you can drop your question in the chat box.
Hello, Dr. Seema. Myself, Sonia. I am teaching microbiology in this department, IHG. I have a question. Can I ask?
Yes, ma'am. Please.
Hello, Dr. Seema. My question is that, as you said, you can introduce biofertilizer on by coating techniques, by dipping roots, or by applying biofertilizer to the soil itself, right?
Yes.
My question is that, how can you use plant tissue culture technique in your research?
Plant tissue culture technique can be used, but yes, ma'am, in case of biofertilizer, we have this way to directly apply. We are not doing any work on plant tissue culture, but these are the plant tissue culture can be used just to see in labs, okay, whether with a particular plant, when a biofertilizer interact with this organism as a biofertilizer interact, how it affects the plant. मतलब कैसे उसको एक particular lab में study करना है, because it is very difficult to study this in pots also and in fields also. तो एक प्लांट के अंदर अगर हम इंटरेक्शन कराते हैं एक पर्टिकुलर बायोफर्टिलाइजर का, तो उसके बाद उस प्लांट में किस पर्टिकुलर वे में चेंज आता है, व्हाट इज द मैकेनिज्म के कैसे वो बायोफर्टिलाइजर प्लांट में चेंजेस ला रहा है, कि वो उसकी उसमें लीव्स में क्लोरोफिल का कुछ मतलब उसमें चेंज हुआ है, या फोटोसिंथेसिस के रेट में कुछ चेंज हुआ है, या फिर कोई पर्टिकुलर हार्मोन की उसमें चेंज आया है कि वो ज्यादा बनने लगा है उस प्लांट में. दैट स्टडीज कैन बी यूज्ड, दैट स्टडीज कैन बी done through these tissue culture techniques. But yes, in direct application in the pots and fields, there is no use of these plant tissue culture.
So you mean that after biofertilizer effect can be studied, phenotypic effects can be studied?
Yes, yes,
Oh,
yes, indeed.
Okay, thank you, Dr. Seema. Thank you.
Any other participants, please? So I think there is no question. Nice presentation, Dr. Seema, and you have replied very nicely the question to the participants.
Thank you, sir.
So Dr. Kalpana? Dr. Kalpana, are you there? Dr. Kalpana, should we close the session or?
So, thank you so much, Dr. Seema Sangwan, for your enlightening talk. And you have wonderfully explained the uses of microbes as a fertilizer to reach to the goal of sustainability. And you have answered all the questions. I'm sure that our students are benefited with this. Now, I would like to thank Professor Renu Arora, Director IHE, Dr. R.S. Thaka, Chairperson INYAS, and Professor A.M. Deshmukh, President MBSI. He is not with us today. And I would like to thank you, Professor Sunita Agrawal, ma'am, State President MBSI, Dr. Kalpana Nagpal, Coordinator NCR Chapter, Dr. Veda Krishnan, Core Committee Member INYAS. And my thanks are due to all the audience who joined us through Zoom and through YouTube. Thank you once again, everyone. Now I would like to request everyone to switch on their camera so that we can have a group photograph. Soumya, you please click the photo. Sunita ma'am, are you there? Dr. Kalpana, are you there? Yeah. Sonia ma'am. Yes, it is done. Thank you, everyone.
Thank you so
Thank
much.
you.
It was a very wonderful experience to explain our work so that we can aware the students how they can use this and they can understand this concept of biofertilizers. Thank you so much. Thank you so much, ma'am and sir.
Thank you, Dr. Seema, for your wonderful talk. Thank you so much.
Thank you. Thank you.
Thank you.
Thank you all.
Thanks.